I will do a very brief introduction of Kosta Plakas, who's with us today to discuss the article that will look at SNPs that can affect signaling in 5-HT2A, the receptor subtype that is important for many psychedelic actions. And so we know that from the talks we've had in the past year and journal clubs that basically there's a lot of interest in these compounds. Some trials are coming out that look very encouraging across several different psychiatric disorders. But one of the things that's very striking is the fact that there is a lot of heterogeneity in the way that people respond. Basically from minimal response to an adverse response to a very positive therapeutic response, sometimes to the same dose of a drug. And so that smells genetic, doesn't it? As though there's something that actually could be modulating those effects in the realm of perhaps single nucleotide polymorphisms. So we're going to hear a little bit more about that today. By the way, that was a very substantial and comprehensive article, Kosta. Thank you for taking it on, and we appreciate it. All will be illuminated. Take it away, Kosta.
I hope it will be illuminated. My main hope is that you will not leave this meeting more confused. And if you learned something along the way, that's great too, of course. But let me go ahead and share my PowerPoint here, and then we can sort of dig in. And it certainly was comprehensive, especially for a non-pharmacologist. And so you'll hear me lament about that at least once or twice more. Can everybody see my screen?
We can. It looks good.
Awesome. Yeah, let me just can we see the laser pointer too?
I think so. And thank you, Chris Petro, for pushing the record button. We appreciate it.
No worries.
Go team. Okay,
right.
so I'll give you everybody a brief outline for the presentation as it stands at large today. So I figured I'd try to arm us with some tools to better understand the paper. And to do that, we'll discuss briefly psychedelics, what they are, why they might be an intimidating word, but they're not all that confusing or that bad. Before moving into slightly more complex things, so the 5-HT2A receptor, which is a G protein coupled receptor. And I find it hard to disentangle these three items in my mind, so I'll probably present them more or less as kind of one somewhat cohesive block. You'll find the lines between 1 and 2 kind of get blurred. So I've highlighted both on the same slide just to show that we'll kind of discuss them all simultaneously. And if anyone has any questions, comments, or concerns during this part, you can certainly interrupt me. As I've mentioned, I'm not a pharmacologist by training, and so tackling this was a psychedelic experience to a certain extent. And so I'd certainly welcome anything you might be able to offer to complement or support this humble organic chemist and radiochemist learning of the subject matter. But then we'll briefly just mention something about the authors, although they likely need no introduction to this group before we dive into the article discussion. And again, at any point throughout this, you'd like to interject, by all means, go right ahead. We should have hopefully enough time to entertain a lively discussion. So psychedelics, like many of the drugs I'm interested in and the group at large is interested in, these are old drugs. They've been around a while, perhaps not as long as things like opioids, but they are both old and somewhat naturally occurring. So NN-DMT or dimethyltryptamine is a naturally occurring substance. You can find it as a constituent of this plant B-capi, which is apparently found in the Amazon basin. Its use is primarily related to ritualistic and ceremonial cultures and have been a part of lots of different cultures, mostly pertaining to Central and South America. And DMT is a key ingredient in the active pharmacological drink ayahuasca, which is reported to be somewhat sacred in certain religions or cultures. And for many years, that was sort of all we knew about it. It wasn't until fairly recently in the grand history of time that these were introduced to the Western world. And it's generally reported that Dr. Humphrey Osmond sort of introduced psychedelics and coined the term psychedelics and introduced them to the Western world sometime around the 1950s. And from the 50s to the 70s, this wasn't a time I was around, but I've heard it was a pretty good time. Like there was lots of open science regarding the development and the use of psychedelics in in medicine. However, that came to a somewhat abrupt stop in the 70s when these molecules were scheduled, and they were scheduled as Schedule 1, meaning that they were dictated to have no medicinal purpose. And so research kind of came to an abrupt halt for quite a long time. It's not until a little bit more recently, and this is a time frame that I'm a little bit more familiar with, the 90s to present, mostly because that's the period of time I've been alive. But from the 90s onward, we've kind of had a resurgence in psychedelic research. Some things a little bit more scientific than others, some peer-reviewed, some not. But some I'll just draw attention to is some interesting work done by Alexander Shuglin and Anne Shuglin. And I bring this up because this is something Brian Roth mentioned in his talk. So this is I don't believe it's a peer-reviewed study on phenethylamines and tryptamines, but it is an SAR study nonetheless of how different structures of these molecules can produce different subjective experiences. And then moving more into the realm of peer-reviewed, legitimate scientific research, the gentleman that we have here is Jason Wallach. And he is a professor, I think assistant professor of pharmacology at St. Joe's. He gave a talk to our group a little over a year ago, detailing some of the structure-activity relationships of psychedelics acting on the 5-HT2A receptor through the use of a technique called SAR by NMR. So using nuclear magnetic resonance in conjunction with synthesis to kind of predict the ability of new molecules to exert an effect on the 5-HT2A receptor. And then in Jason Wallach's group, there's a gentleman known as Hamilton. So Hamilton Morris is I guess a science journalist. And so he has a pretty interesting, I think it's a Hulu series where he travels the world and, studies different psychedelics and non-psychedelics and, their cultural significance. And he also tries a couple of them. So I think there's the disclaimer, kids don't try this at home. And so that goes for, both the PCAL, TCAL, but also Hamilton's Pharmacopoeia. It's definitely interesting to kind of see how psychedelics have captivated the community and sort of where they've pushed clinicians and journalists into. So psychedelics, it's definitely a term that has gained more prevalence in lots of literature, not just scientific. And so I think it's important to remember psychedelics are just small molecules that interact with a certain receptor. So psychedelics act on the 5-HT2A receptor. It's shorthand for the 5-hydroxytryptamine 2A receptor. And the psychedelics are small molecules that can generate perceptual distortions, psychological experiences, or labile moods. They can be classified into tryptamines or phenethylamines. And so tryptamines, which we have here on the left, have this fused 6,5 membered ring, and then a 2-carbon spacer, and then some sort of a nitrogen bearing either hydrogens or methyl groups. So these are, again, the tryptamines. There's also phenethylamines, which have this phenyl ring, a 2-carbon spacer, and in this case, the NH2 moiety or functional group. There's also a third called the ergolins, of which LSD is a member. And these are interesting. I think they can kind of fit into both classifications. So if you look carefully, you can kind of see highlighted. They have that DMT-like scaffold highlighted where you have this 6,5 fused bicyclic system, 2-carbon spacer, then a nitrogen. But they also have this phenyl group with 2 carbons between the nitrogen as well. So it kind of straddles the line. And in some cases, ergolins exert characteristics that might be more representative of tryptamines or phenethylamines. And it should be of no surprise that these are structurally similar to endogenous neurotransmitters such as serotonin, which you'll hear me use the term 5-hydroxytryptamine and serotonin interchangeably. They're synonymous. And the phenethylamines kind of have a resemblance to dopamine. So signaling at the 5-HT2A GPCR is mediated by psychedelics and some endogenous ligands too, but an agonist in this scheme denoted by a star will bind the 5-HT2A receptor, and this can encourage a variety of different downstream behavior to happen. So upon agonist binding these GPCRs, the 5-HT2A receptor can either undergo beta-arrestin recruitment, which has one sort of distinct set of outcomes, or it can engage in G protein mediated signaling. which summarily will result in the release of cortisolic calcium. And a somewhat hot area of research is studying how the bias of an agonist can either change as the result of a single nucleotide polymorphism. And that's sort of the crux of this paper. Also, the design of agonists that preferentially activate one pathway over the other is equally as exciting, and will have some more implications that I'll touch upon a little bit later. In going through this literature, I was somewhat surprised to see that PET ligands had sort of been developed for the 5-HT2A receptor and characterized quite well. So this is a group, I believe, from Amsterdam who had done a fair amount of SAR designing these carbon-11 CIMBI or SIMBI ligands, SIMBI or CHIMBI. And so these ligands have been radiolabeled carbon-11. SIMBI-36 was the best performing radioligand they had developed, and perhaps not surprisingly, it shows uptake in the frontal cortex, the cortex, and I'm told these are regions of the brain in which you are most likely to find the 5-HT2A receptor. So this is SIMBI-36 is a pretty potent single-digit nanomolar binding radioligand that has subsequently been used to characterize psilocybin and its metabolites receptor occupancy, which is shown here. So the same group used SIMBI-36 along with some pretty standard fare PK/PD measurements to correlate the plasma concentration of psilocin, which is the key metabolite of psilocybin, as a function of time. So these are human subjects that have ingested psilocybin. And you can see there's a pretty nice relationship between the amount a patient would be administered and the plasma peak concentration. There's also a bit of a time dependence. It takes about 60 minutes or so to kind of reach peak plasma intensity or peak plasma concentration. This correlates somewhat nicely with a Likert scale, which the patients were asked to fill out. So the Likert scale correlates with intensity, with 0 being not intense feelings, or 10 being intense subjective feelings of the reported trip that they would undergo as a result of their ingestion of psilocybin. And then using the PET tracer that I described, they were actually able to quantify receptor occupancy, correlate that with intensity rating based on the Likert scale as well, and then further correlate that to plasma psilocin concentration, showing that indeed the higher the receptor occupancy, the more likely or the higher rating patients kind of scored on this Likert scale, suggesting that there's a correlation between how much a patient would ingest, the plasma concentration, and the subjective experience they may be experiencing. So all of this is more or less standard fare for drug discovery, right? So we have a molecule that's interesting. Maybe it comes to us from nature. Maybe it's something we've synthesized in lab and has exhibited some interesting properties. And so all of that, again, pretty standard fare. But what I think sets psychedelics apart is that the clinical effects can be long lasting and they can be quite profound. So these are just some snippets that I've taken from a paper by Matthew Johnson, who should be he's not a stranger to us. He's presented somewhat recently in Roland Griffiths, who's another kind of powerhouse in the field of using psilocybin-assisted treatment for not just major depressive disorder, but also substance use disorder as well. And kind of what stuck through me upon reading some of these reports is that I'll just read the quote, is that these findings demonstrate that the substantial antidepressant effects of psilocybin-assisted therapy may be durable at least through 12 months following an acute intervention. And so that seems to be kind of echoing or rather pushing a paradigm in psychiatry to leverage substances with quote unquote acute psychoactive effects to induce long-term benefits. And so I know a couple of drugs, but I don't know many that do this, and that sort of runs counterintuitive to what we might think of as being a viable treatment as a medicinal chemist, right? So generally, we want to make something that we can dose once a day. There's certainly, I think, more money to be made that way. Not that that's a good thing, but that said, I think a major reason that we don't see a lot of large pharmaceutical companies sort of putting their hat in the ring for this is that there's not, a patient population that can be dosed repeatedly, and that's problematic in and of its own sense. I just wanted to mention that these are indeed kind of disruptive. They're new, and I don't know many molecules that sort of possess this property. That said, as Anna Rose alluded to in the opening and in the email she sent out, this is good news, right? We can get long-lasting benefits with an acute intervention, but response, both clinical and subjective, to these psychedelics is not uniform across the population. And I think that's the question that Brian Roth and colleagues sort of start to set out in their reason for undertaking this study. So this is these are members of the Brian Roth group. Gavin Schmitz is a MD PhD student. Couldn't find his picture, but through LinkedIn and the Roth Lab website, I was able to find some information on Dr. Sam Slocum, who's a postdoctoral fellow at UNC, and Dr. Manish Jain, similarly a postdoc. And again, these are both postdoc fellows in the Roth Lab. So Dr. Brian Roth is an MD PhD. He presented, I think, to our group at the end of last year, if I'm not mistaken. I think it was fall, but Brian Roth's a pretty well-known powerhouse in GPCR pharmacology and psychiatry. I should also give a shout out. He's the showrunner for the PDSPs, for the Psychoactive Drug Screening Program, which is a pretty neat resource that I use to submit a whole bunch of the ligands I make to determine their affinity and intrinsic activity at various receptors. So if you're an NIH-funded investigator, I think you could submit your ligands here and kind of get the screening done for free, underscoring the role that his research plays in ligand pharmacology. So with the background kind of fleshed out, let's jump into the article, unless there are any questions or comments. And again, you can shout and interject at your leisure as we move more and more into the area outside of my comfort zone. So we'll start kind of just with the basics. I mentioned the 5-HT2A is a protein. It's a GPCR. So in the coding of these proteins, some things can happen. And so SNPs can happen, or non-synonymous single nucleotide polymorphisms. And that occurs when there's a change in the nucleotide in the genome that results in the change of an amino acid residue in the protein. And so there are 7 that are somewhat common. And for a SNP to usually count, it usually has to affect, I think, greater than 1% of the population. So these are 7 SNPs that can happen, and by that definition would be present in greater than 1% of the population. That can happen at the 5-HT2A receptor. It is worth noting that these SNPs appear to be somewhat removed from the active site. So these are going to cause some issues in ligand binding, but these are going to be far removed from the orthosteric site. And I think that's interesting. So it's hypothesized that the subsequent change in efficacy and potency we will see will likely have to do something with conformational stability of the G protein.
I just had a quick question, because this is certainly outside my usual zone, but did they deliberately select things that were remote from the orthosteric binding site? Is that because, I don't know, it seems like the closer to the action you are, the bigger the impact you might have, naively. So anyhow.
That's a good question. And my first thought on that is that SNPs closer to the orthosteric site might present particularly bad outcomes. So I don't know if someone with that would be able to make it to a clinic to have like either their genome sequenced, or I'm not sure if
Yeah,
that's
I don't know either. I'm just curious about whether that's a general principle, because if it's something that's so close to the hotspot that you are concerned with affecting it too much,
right.
And finding a sweet zone where you can actually affect it, but not in a way that would be deleterious or life-threatening. Anyway,
Right.
Just a passing question. So thanks.
Sure. So excellent comment and observation. And what I found comforting that the authors provided was that they first tested the potency and the span. So I'll use a couple terms interchangeably. When I say potency, I'm generally referring to the EC50 or negative log, the PEC50, and span I'll usually use to refer to either the efficacy or Bmax. And so what the authors will test first here is the potency and the span, or the EC50 and the span of the endogenous ligand, so 5-hydroxytryptamine, at the various SNPs. So they'll test it at the serine 12 SNP, the threonine 25 SNP, and so on and so forth. And what I found kind of comforting and reassuring is that all of these SNPs do not cause a significant change in the potency of 5-hydroxytryptamine mediated signaling. So all of these, in theory, will have the same EC50 value if you were to kind of trace them or draw them or otherwise calculate them. So no statistical difference between the measured PEC50 values of the wild type and SNP containing receptors. However, 2 or 3 of these SNP containing receptors, so 3 of them, do possess a difference in span or efficacy. So the serine 12 ASN SNP, the isoleucine 197 valine SNP, and the histidine 452 tyrosine, they have changes in their efficacy or Emax, if you will, or span, right? So, and you can kind of pick this out just by looking at it. So the serine 12 one will be here. It looks like the span or the efficacy is actually elevated to somewhat of a superphysiological level. And so they measure that if your wild type receptor is at about 100%, your serine 12 polymorphism encourages a bit of a, an above normal signaling efficacy, whereas the isoleucine and histidine SNPs kind of drop that. And you can kind of pick this out again visually, right? So this is dropped significantly below where we would see the wild type receptor signaling, and same for the histidine 452 tyrosine SNP as well. So that in and of itself might cause an area for concern, and we'll see again how the psychedelic signaling will change as a result of these, and that might underscore different subjective or clinical outcomes. So as I alluded to, select SNPs do cause changes in psychedelic-induced efficacy or EC50 values. So if we focus on psilocybin, or in this case, they use its metabolite psilocin, we can see that the efficacy of psilocin for the wild type receptor is about 5 nanomolar, but as the result of either the ALA-230 threonine SNP or histidine 452 tyrosine SNP, the efficacy is actually dropped a bit. So it goes from about 5 to about 40 or so, suggesting that the efficacy of psilocin has dropped, or it's become less efficacious or less potent rather. And we can kind of go through this for each set of psychedelic and polymorphism. And we can kind of quantify that and triage that. So similarly, DMT will be, this is actually different. So psilocin loses its potency or becomes less potent, but DMT will actually become more potent as a result of the SNP. So for the wild type receptor, DMT has a potency of about 10 nanomolar, and for the ALA-447 val, it actually becomes more potent. So the potency is now about 3 nanomolar or so. Mescaline is an interesting one in that the potency similarly increases. So mescaline for the wild type receptor is about 4,000 nanomolar, and it increases significantly for the serine 12 ASN, the threonine 25 ASN polymorphism SNP, and also the ASP48 ASN and the ALA-447 val. So as of right now, they all seem to trend in the same direction. Very few of them split in potency. So a polymorphic change does not seem to both decrease the potency of one psychedelic while increasing the other. LSD is kind of boring in one regard, in that none of the reported SNPs cause a change in the potency across any of the SNPs. So its potency sort of stays the same. Now, the other thing I'd like to draw attention to, and I did not a great job of color coding this, but in addition to the potency, the Emax, the efficacy, or the span, again, I'll use those terms interchangeably, but the Emax or efficacy can change as well. And so I've kind of color coded. The light green would mean we have an increase in the Emax or maximum response that the psychedelic can induce on the polymorphic receptor, and red, which shows a little bit better on my screen, indicates a drop in the Emax or efficacy. And so you can see that there's various different things that can happen, but taking, for example, let's look at psilocin at the histidine 455 tyrosine residue. You can see illustrated graphically here, right? So the black is the wild type receptor, and then the we'll call that this greenish blue color. We can definitely see a marked decrease in the Emax, or the maximum response you can get as the result of this polymorphism. And so this, in conjunction with the potency, may underscore some of the variable pharmacodynamics that you might see in a patient population, and also underscore different subjective but also clinical responses. And so, as I alluded to kind of in the beginning, there are at least two things G protein coupled receptors can do. They can engage in G protein mediated signaling, which is what we've just discussed. But they can also recruit beta arrestin, which is another downstream signaling pathway. And so what the authors did next was to quantify beta arrestin 1 recruitment. And we can see that there is no significant change as the result of these single nucleotide polymorphisms. And so, for example, let's look at psilocin. Psilocin will have some sort of response or some sort of potency that it engages or encourages the receptor to have, but that's more or less similar kind of across this population for beta arrestin 1 recruitment. Where we start to see some interesting changes, though, is in beta arrestin 2 recruitment. So that's another way in which these G proteins can sort of undergo signaling. And so as the result of some of these single nucleotide polymorphisms, we see changes in the this would be potency and recruitment of these downstream proteins. And again, that's as the result or as the function of different SNPs. Does anyone have any questions or comments on this before we move into bias?
Not knowing enough about this this arena in terms of fiddling around with these polymorphisms and not having done enough background reading, do you know why they chose the SNPs that they're examining? Just in general? Is it because there was already a literature?
That's a good question, and that would be my assumption. So I'm fairly certain that different clinical outcomes have been seen for both psilocybin and LSD before. I don't know if those studies had reported on looking kind of retrospectively at what SNPs might have caused them, but that might be a good place for me to start and discerning why they specifically chose these SNPs. Certainly, if there is a 5-HT2A pharmacologist on the call that would like to chime in, I would welcome your contributions as well.
Well, we'll toss the question up there and somebody may have a thought.
Yeah, sure. And again, always welcome to kind of join in and interject at your leisure. But I'll move on to what I think is a little bit more interesting, and that's bias factor. And so these SNPs, as we mentioned, is kind of the theme of this paper, cause or can cause changes in either the G protein signaling pathway or the beta arrestin signaling pathway. And quantifying that change is interesting and can perhaps be used to kind of triage or select patients that might be or might have benefits from engaging in treatment with one psychedelic versus another. So what the authors did next was quantify something known as bias factor. And so this is in essence a change in the ratio of EC50 to Emax or Emax to EC50 in both the G protein mediated pathway or the G protein signaling pathway to one of the beta arrestin pathways. And so they compare this separately for the beta arrestin 1, or rather the G protein pathway versus the beta arrestin 1, and G protein pathway versus the beta arrestin 2. And again, compare that to signaling at the wild type receptor. So psilocin and psilocybin will elicit some sort of response, which is kind of represented some sort of G protein and beta arrestin 1 and G protein and beta arrestin 2 response, both of which are represented by these dashed lines. And then the SNP will sort of alter that. It'll change the relative Emax to EC50 or not. There's not always a significant change. Sometimes there is. And so what we can sort of tease out from the analysis of the plots on the previous page as represented here is that, let's take psilocin, for example. Psilocin becomes, or psilocin signaling becomes more active in the beta arrestin 1 pathway as the result of the serine 12 ASN SNP versus the wild type receptor. Similarly, it becomes more active in the beta arrestin 1 pathway as the result of the ALA 447 valve SNP. Conversely, it becomes more potent in G protein mediated signaling with the histidine 452 tyrosine SNP. And then we can do the same comparison for psilocin in either the G protein or beta arrestin 2 pathway. So there aren't many significant changes in bias as the result of these SNPs, but it does become, psilocin becomes more biased or more active in the G protein mediated pathway as the result of the ALA 233 SNP as shown here. Similarly as the histidine 452 tyrosine SNP as shown here. And you can do this for all of the variety of different psychedelics that have been sort of tested in the paper and kind of tease out what the effect of a SNP is on the bias or the change in signaling efficacy to potency ratio. And again, they've sort of done this for all of the SNPs and psychedelics examined here within the paper. I mentioned I would hint at this previously, and so biased signaling and biased agonists are kind of a hot area of research, and I think are certainly of relevance to my research, and also the P30 as well, kind of at large, where either designing small molecules that can engage in bias signaling, i.e. activate one pathway like the G protein pathway over another, or looking at SNPs that maybe have higher activation of one signaling pathway over another. It's interesting because you can sort of look at how that might dictate clinical outcomes. So, for example, with opioids, mu-opioid receptor agonists, there's kind of a hot push to design agonists that can kind of activate this G protein mediated signaling pathway over beta arrestin 1 and 2 recruitment, because it's thought that G protein mediated signaling is by and large responsible for analgesia, whereas beta arrestin recruitment might be more responsible for bad things like respiratory depression. And so, again, just something I kind of wanted to bring to everyone's attention as a hot area of research. And I think this is an excellent use of quantifying the response each agonist will cause as a function of different single nucleotide polymorphism.
Kosta, did you go through some of this with your antagonist that you're developing for the mu-opioid receptor in terms of trying to select for or tweak toward a bias for avoiding beta arrestin and going in the other direction?
I think that will be something I do in as I develop delta opioid receptor agonists, which may engage in beta arrestin and G protein signaling. For the agonist, for the antagonist is I've just ensured they're non-activating, and so we use a similar.
And that's all you have to do. Yeah, with antagonists, it's less of an issue. Obviously.
Exactly, a little bit more user friendly or medicinal chemist friendly.
Sure.
Those assays are a bit more easy to read from the Roth lab, but I have some data I'll certainly be happy to forward you.
That's fun. Yep.
Any ideas about where side effects come from, like flashbacks or
psychotic reactions.
Well,
Which cut them off the market, basically, you know.
if we're talking side effects about opioids in particular, I would throw my hat in the
5-HT I
ring.
think he means
I know that-
5-HT2A.
Yeah, I was afraid you were worried about that.
Yeah.
I don't know if I could say that the same side effects, or rather the side effects are caused by activation of the same
Yeah.
pathway in each.
Different systems, yeah.
Exactly.
Well, one of the things going to 5-HT2A, and I think David Olson and other people are working with these compounds that may not have an hallucinogenic impact, right? By using the head twitch screen and so forth and so on with the 5-HT2A. So it may be, George, similarly, possible to select for compounds that have fill in the blank therapeutic effects, but are less likely to have hallucinogenic properties, which of course, it's an open research question about whether or not they may be correlated with benefit, but is that a correlation? Is it necessarily causal, or could you get by without it? Because of course it makes therapeutics much more challenging if you have something that's a full-on trip. So, but the same idea, I think that's what you were getting at, is it possible to select around that? And in fact, that is one or two career paths that I know of that people are following. Yeah.
Yeah.
Go ahead. I'm sorry.
No,
Kosta.
thank you for the contribution. And I will add it. It is nice that these tools are, certainly a little bit more readily available now. So hopefully we can have this at our disposal, should we want to, or should somebody want to do the experiments you've just described. So select for something that has, a real clinical benefit without, perhaps the deleterious side effects. So we're just a couple more slides to get through before we can maybe get into a proper discussion. But the other thing that's worth mentioning is that these psychedelics can change their potency over time. So these Brett assays were done and either quenched at different times or otherwise analyzed at different times. And so what these have shown is pretty interesting is you can get either an increase or decrease in signaling potency over time. Which I think is a bit unexpected, at least given my somewhat limited knowledge of the literature. So if we take, for example, a look at psilocin, psilocin will have, a potency of about 27, and then it will become more potent over time throughout the course of about 180 minutes. DMT will more or less kind of do the same thing, maybe not reaching the same levels that psilocin will. Mescaline, again, similarly follows a similar trend, and this is also illustrated graphically. Psilocin I found to be interesting in that, while it does demonstrate increase in potency of the G protein mediated pathway, it shows the opposite. So a decrease in potency for the beta-arrestin 2 recruitment, and kind of across both groups. I think that's the only psychedelic that does that. So just something I kind of found interesting, perhaps underscoring why it has been, I think it's the most prevalently used in clinical trials to date. And I have a supplemental slide I'd love to get everybody's opinion on in a couple slides, but just to highlight that, the signaling can certainly change in potency over time. LSD is interesting in that it can show quite a drastic change in potency. So that's also illustrated nicely by these colorful graphs. So we have signaling at 5 minutes, and then we notice quite a large shift in potency going from 5 to 30, all the way up to about 300 minutes. I mean, that's illustrated numerically here. So the potency of LSD for the wild type receptor is about 6, and then it actually becomes several orders of magnitude more potent, which is interesting. Here, beta-arrestin 2 follows the same path, in that it becomes more potent as well. So starting with this black line here, you can kind of see it's sort of pushed to higher potency as a function of time, which is in contrast to psilocin, but in the same line of in the same trend as DMT and mescaline. And also that is perhaps should come as kind of no surprise that SNPs can also induce the changing over time as well. So the ALA 233 SNP exhibits a significant change compared to the wild type signaling after 5 minutes. And that change, though exhibits the same trend, it is much more potent initially compared to LSD for the wild type receptor. The authors also quantified a receptor density, which I think is important, just to make sure we're not, dealing with orders of magnitude more or less receptor as a function of single nucleotide polymorphism. And so they do note that there is a difference, and these are saturation binding assays, which they've used proteated contensorine to quantify the amount of protein both in the wild type and all of these single nucleotide polymorphic receptors. They mentioned, yes, there is a difference. They also mentioned that Brett-based assays can be relatively insensitive to receptor density. I think that's a convenient conclusion to make. I don't know if I buy that one way or another, although I should note I'm relatively inexperienced in both performing and analyzing these tissue-based saturation assays to quantify receptor density. And so with that, I think I'll quickly try and wrap up the big picture conclusions as generally being something I'm more comfortable talking about. But the authors note that individuals carrying this ALA-233 SNP variant might show altered responses to the psychedelic studied, and that's given that either the G protein mediated and or beta arrestin signaling was changed, and that's illustrated numerically here, where compared to the wild type receptor, we have a change in potency for psilocin, and we have a change in the beta arrestin signaling for DMT, mescaline, and LSD, again, compared to the wild type receptor. Individuals carrying this His452 tyrosine variant might show additional altered responses to psilocin and psilocybin, as there's a change in G protein mediated signaling, which can change signaling bias, right? And so as a function of the SNP, we see a decrease in potency of psilocin for the receptor. So that could perhaps drive a change in signaling bias, which might cause a difference either in subjective or clinical outcome experience as well. And additionally, in this polymorphism, we have a change in the efficacy or Emax, as I've tried to color code here. And so that might lead you to believe that maybe triaging patients based on presence or absences of SNPs might be a good idea, something worth doing, and something I'll sort of pose to the group in a slide or two. I will mention that I did have one maybe limitation to bring up, and I'm biased as a radiochemist, that I thought it was kind of hard to correlate the in vitro or this in vitro study, though a real powerhouse of pharmacology. I found it difficult to see if there was a correlation between individual perceived intensity and the presence and or absence of SNPs. I don't think that was within the context of this paper. So that's, kind of take what I'm saying with a grain of salt, but I still think it would be interesting. I'd like to know more about correlations of clinical outcome with the presence or absence of certain SNPs at large in patient populations. And so we can kind of open it up for a discussion. There are just some, maybe interesting observations I had. One thing I found interesting is that I wanted to know if there was a correlation between variability in either potency, efficacy, or bias as the result of changes in efficacy or potency as a result of SNPs, and how that correlated with how many drugs are currently in clinical trials. So based on the paper, there are about 5 changes in bias that are present in the population when using or when measuring it using psilocybin, but there's still quite a large number of ongoing clinical trials using psilocybin. So I guess I wonder, what the relative percentage of these SNPs is in the population compared to relatively small numbers of clinical trials ongoing with DMT, but there are, less changes in bias as the function of changes in potency and efficacy. Same thing for LSD, only 2 changes in bias, but still, only a fraction of the number of clinicals that are being undertaken compared to psilocybin. Yeah. So with that, I'd be happy to engage in any conversation, hear your thoughts. We got about 15 or so minutes left.
Thank you so much, Costa. And I'll jump in quickly and then I'll shut up and let other people chime in. But I just wanted to say, I think this is so valuable sort of as a template for this very early stage in the field where I think, and thanks to Anthony, there's a lot of SNPs for 5-HT2A receptors, about 255 he found in a quick look at the literature. And Brian Roth and they chose these to study. I think it will be incumbent on us with the imaging studies to, it's cheap to get a blood sample, get the DNA extracted, and then with relatively low cost, probe for some of these SNPs that are shown in work like this to have potential for modulating effects at the human level. Because if we don't, and we find that we have a lot of heterogeneity and we aren't balancing for it or we're blind to it, PET is very expensive and the samples are always much smaller than what you would like. And so we would be at the mercy of that kind of heterogeneity, so that we would, the fantasy would be, and this is something we've talked about in terms of potential P30 pilots, is to be able to do a PET scan before actually doing psilocybin or other psychedelic administration, and based on what we see in terms of the person's response and their 5-HT2A receptor availability, and now we can add their SNP profile, whether or not they're more likely or less likely to benefit or have a negative or adverse consequence. And it could guide research in a way that would be really helpful, given the very long delay in any of this work, to be able to actually do it in some rational way going forward. Otherwise, we're going to have lots of small trial studies, small sample sizes with lots of heterogeneity. Some will look promising, some will look underwhelming. And before the field gets disenchanted with the fact that it doesn't seem to work for everybody, there may be some really good science to bring to this so that we can understand why. Anyway, that's my pitch for trying to add a receptor level measure at least to these trials. And at least if you can't do receptor level stuff because you don't have a PET center, you can at least do psilocin levels, because they actually correlate pretty well with the therapeutic benefit, for at least, I think, with the intensity effects, I believe I saw in at least some of the clinical work. So it's not the same thing as having a receptor level measure per se, but it showed some pretty good correlations. I should look back at that. Anyway, enough. I'll shut up.
I can just make a comment, because I was one of my supervisors as a resident had participated in trials at Fort Detrick, Maryland, that the Army was doing using, I think it was LSD as a biological warfare agent. And I think the Army got in some trouble about that because they were having some clear effects. It's, I just read the literature. I've always been interested in this little group, but always with a lot of caution, because
well, I think it was one of the things that actually impacted the research in the field, right? Is that the government was doing some experiments, perhaps, without all due caution, and sometimes without consent forms that were very clear about what
that's how they got in.
And so, I think it really had a huge impact on the field, and essentially set it back for some time. That, along with the fact that these sort of became, street drugs that had a colorful reputation, but they no longer were taken seriously as potential therapeutics. And so the combination of those things has really sort of held the field back. And I'm hoping that things can proceed sort of rationally.
No, I think that's absolutely true. It's very great that a lot of all this work's going on, looking at the biology, because there seem to be some clear therapeutic effects with some very tough cases too. But it's always struck me as a really tricky business. So
it's tricky, but that's why my fantasy, George, would be that if you had some good biologic markers, that you might be able to know who's likely to benefit or less likely to get into trouble even before you actually administer the substance. And that would be really helpful. That's down the pipe, but I think that would give you very, very powerful therapeutic possibilities then.
Right, of course, yeah.
But can I just, so this is really a question and a comment. So, I mean, looking at some of these propensity for the SNPs, it's incredibly low in the population, right? I mean, 0.003%, and even sort of the higher one. I thought it was interesting on the graph you showed, Kosta, where the pink, which had lower effect, was actually the one SNP that was, high. Yeah, right. This one. So the histine to tyrosine, that has the highest proportion, but it's like only 8% in the population, according to the paper. I was just curious, any ethnicity known about these SNPs in terms of, are they more prevalent? And, to Anna Rose's thing, would you even in a sample size of 20 or 30, would you ever even get someone with any of these SNPs?
That is a great question that I wish I had the answer to. No, yeah, I realized that the percentages were probably low, and yeah, in a small sample that might not be recapitulated. That said, I don't know if there's any different geographical regions where these may be more or less prevalent.
Any of our geneticists on the call that know anything about the major and minor allele frequency for some of these things, that maybe even the ones that Brian Roth didn't test, but that are begging to be tested? If not, it's something for us all to look up later.
I have a question. First of all, thank you so much, Kosta, for the presentation. It was really, you did a really great job of sort of explaining the research and the findings. One thing I was wondering was, it seems like there's this pretty clear evidence that there's sort of a variation in the pharmacological response to things based on SNPs in this gene. But I was wondering, has there been any literature to sort of link genotype in general, either with these SNPs or in general, with the heterogeneity that's seen in the actual response, like in terms of like the treatment efficacy and the subjective effects that people experience?
Yeah, there are a couple of papers listed that I've sort of made note to go back and check out. But yeah, they mentioned, I think in the introduction, that one of the drivers of this paper is that there has been a mixed efficacy or mixed response to psilocybin treatment or other psychedelic treatment. However, I don't know if that, I don't think that's tied back to like the phenotype yet. So there is difference, and I think this paper was trying to address why. And I think it's a pretty solid hypothesis that you can get different responses based on the polymorphisms, but yeah, to my knowledge, it's not formally tied together just yet.
Yeah, it looks like there's a lot of good room for research, Yusuf. Try to frame it in a positive way, that we're at the very early stages, but that this does hint that there's something to look at.
Thank you.
Anyone else? We still got 5 minutes.
I just wanted to say, if you want to, you have presenters option to give a 1-minute update on your work that you've done with Brian Roth. You sent out a whole bunch of antagonists for the MOR, right? And basically he did essentially bioassays and behavioral assays to tell you whether or not they were functioning as hoped.
Yeah, yeah.
A word about Brian Roth, who is a powerhouse, he is the NIDA guy who has this basically lab where, as Kosta says, if you have an NIH grant and you have compounds to be screened, he's the man. Do you want to say just a moment about that?
Yeah, I will say his lab is the powerhouse. So you can, if you're an NIH funded investigator, you can ship your small molecules there and request for them to be screened for potency. And so you get various different KI values. You can screen for intrinsic activity. So classify them as agonists, antagonists. I don't think they can do inverse agonist yet. talk about that later, but I've sent a whole library of small molecules there. Designed as small molecule mu opioid receptor antagonists. We have some interesting and encouraging data back that they are potent, they are selective, so that's good. And most importantly, they are antagonists. So
Yeah.
the key advantage of a potent selective antagonist is that it could perhaps replace carfentanil as a mu opioid receptor PET ligand. Carfentanil, which has a very strict specific activity requirement, because it is so potent, you can cause respiratory depression even at tracer doses. So the antagonists generally have
Yeah.
better
Yeah.
safety profile.
Yeah, carfentanil was, a bear to work with even before everybody's sort of now with the fentanyl-driven pandemic, very aware of the potency of fentanyls and carfentanil even more than just the quote unquote regular fentanyls. But to have antagonists that are very safe to work with, you do not have to have naloxone at your side if you're working with an antagonist compound. You
Right.
basically have a lot of freedom, and for certain kinds of questions, if you want a really nice, highly specifically high affinity bound antagonist that does not have sensitivity as much to endogenous competition, from endogenous 5-HT, then it'll be wonderful. I'm sorry, endogenous opioids. It would be wonderful to have these available, and we just really haven't. So we're thrilled.
I'm thrilled
And
and
I think,
excited.
and I think you're getting close to writing a grant around some of these.
Yeah, maybe that might be something we should also discuss offline. I would like to write, I guess Bob tricked me into writing a K. Bob, if you're on the call, sorry. But yeah, no, it's going to be mostly delta opioid driven. So I have a whole library of delta opioid agonists, for which an agonist for the delta opioid receptor is much safer than a mu opioid receptor agonist.
Sure.
No analgesia, less respiratory depression. If anything, has a bit of an anxiolytic effect. So maybe framing that for something down the road.
Well, yeah, you can write as many grants as you want, Costa. We'll look forward to them all.
Oh boy. If I write a
But,
bunch, maybe one will get funded.
Yeah. But just, and I will put in a word, I mean, obviously for your antagonists, for your MOR antagonists, obviously one of the things people are looking for are things that might be helpful in reversing the fentanyls. Regular old naloxone sometimes has to be given multiple times.
Sure.
And so having something that's very high affinity bound, longer acting, has better pharmacokinetics. So some of these things that are being developed as tracers may also have a future life as very important anti-overdose therapeutics. So just saying, I think you're going to be a very popular person.
Absolutely. That is the goal, kind of killing two birds with one stone.
As you will recall, Anna Rose, from our P30 call last week, the mu antagonist grant was just submitted a week ago,
Great.
the R01. So that's going to be under review in the fall, but Kosta is taking the lead on the Delta project.
That's good. All is good. We'll all look forward to it. And thank you, Kosta, for doing a really good job with the presentation today, giving good background for those of us who need to be lifted up and pulled along on these topics. We appreciate it.